We're going to spend some more time today talking about osmosis, and I want you to make sure that you have your coloring sheet on passive transport available. I do want to make just a real quick mention of the diagram that's on the top, because to me the way this diagram is drawn, it's a little bit confusing. This triangular area on the bottom of this first picture, that is supposed to represent a chunk of whatever solute they have put into that solution. So if it's a chunk of salt, a chunk of sugar, a chunk of whatever it might happen to be, that's supposed to represent that chunk, and then that chunk dissolves and then everything gets evenly distributed all the way throughout. Now, I'm not going to spend any more time on that diagram. What I really want to focus on are the three diagrams here in the center of the page, because they are relating to osmosis and the three different types of solutions that you can put a cell into. I'm actually going to go ahead and put a chart on the board to help you differentiate between these three different types of solutions. So the first one is isotonic. And an isotonic solution means that the concentration is the same both inside and outside of the cell. Now, concentration just means how much stuff is dissolved. So if you think about, you have maybe 2 glasses of water, and then the first glass of water, you just take a salt shaker and you just shake a little bit of salt into it. Whereas the second glass of water, you take the cap off the salt shaker and dump all of the salt into it, and then you stir them up and get the salt to dissolve. Well, in the first glass, your salt is not very concentrated compared to what's in the second glass. The higher the concentration, the more stuff that's dissolved in there. That's all concentration means. So if I have an isotonic solution, that means that the concentration of whatever it is that is dissolved, whether it's salt, whether it's sugar, whether it's who knows what it might be, whatever that concentration is, it's the same both outside the cell and inside the cell. So there's no concentration gradient, because there's no difference between the concentration inside and outside the cell. And like we talked about the other day, we said that water is going to continually move across the membrane in a very random fashion. So the water is going to move both directions. So the water moves both directions across the membrane, but there is no net movement that's going to occur with the water. You're going to have just as much on one side as you do on the other side. There's not going to be a big change, okay? So if you look at the pictures here, the very first picture, you can see that on the side here it says water, and they have arrows going in and arrows going out, and the arrows are both the same size. So what that tells you is that the water is going in and out at the same rate, both in and out, same rate for both of them. And then all of these dissolved particles, it kind of looks like, I didn't count them or anything, but there's approximately the same number of particles inside as there are outside. So that's talking about the piece that is dissolved. The second type of solution that we need to talk about is hypertonic, a hypertonic solution. And in a hypertonic solution, the concentration outside of the cell is much greater than the concentration inside the cell. So outside is greater than inside. So think about what we talked about before. What does that mean? Well, if you think about the dissolved particles being invaders, and the water is going to go try to help shield the other guys from the invaders, if you've got a lot of concentration of stuff outside the cell, the water inside the cell goes, they're in trouble, they need help, and they start going outside the cell to try and diffuse the effect of all of the invaders that are outside the cell, if they're viewing the solutes as invaders, okay? So what happens is that your water then is going to move out of the cell, because my heading here says water moves out of the cell in an effort to equalize the concentration. There's a whole lot more solute outside, so the water moves outside of the cell to try and make things equal. Well, if you have too much water start moving outside of the cell, what happens? Well, eventually the cell starts to shrivel because all of the water is moving out of it, and when the cell shrivels, that's called plasmolysis. Okay, so the cell basically just shrivels up. That is what the second picture on this diagram is supposed to be representing. Here you can see that the water is, the arrows are all moving outside. The cell is supposed to look like it's shrivelly because there's a whole lot more particles on the outside than there are in the inside. So the water's moving out to equalize the concentration and the cell just shrivels up. Okay? By the way, that is what would happen if you tried to drink ocean water, because the ocean water has a whole lot more salt in it than the cells in your body do, and so all of the water starts moving out of your cells to try and neutralize the salt water that you drank, and you just get thirstier and thirstier because your body's craving more moisture to try and get that into your cells so that they can maintain homeostasis. Okay, so what I want you to do before we go any further here is take your pencil, and under this first picture I want you to write isotonic, because this cell is in an isotonic solution, water's moving equally. And then under the second cell I want you to write hypertonic, and then I want you to write plasmolysis. So write hypertonic and then write plasmolysis, so that you have a picture in your head of what that process looks like. Okay? The third type of solution is hypotonic, H-Y-P-O, tonic, and what happens here is that the solution, or the solution outside the cell has a much smaller concentration than what's inside the cell. Okay, so this time our symbols turn the other direction. And so which direction is the water going to move now? Well, again, there's more invaders the other direction, so now the water decides it wants to flow into the cell to try and help the water molecules on the inside. And so the water is going to move into the cell in this particular case. And the water moves into the cell, it moves into the cell, it moves into the cell. Eventually, the cell, try to say that 10 times fast, the cell starts to swell. There we go. And it swells up and it swells up and it gets bigger and bigger, and eventually the cell can actually rupture and burst. And that is called cytolysis. When the cell gets so much moisture, so much extra water in it that it actually ruptures. And so that is what this last picture looks like. On this side, you can see that the water is moving into the cell by the directional arrows. You can see that there's a whole lot more solvent particles inside than there are on the outside. And so the more water rushes in, the cell gets bigger and bigger and bigger. You can see that this cell is much bigger than it was here at the beginning. And so this cell eventually is going to swell up and rupture. Okay, so cells have ways that they can prevent this rupturing. It's quite a bit harder to prevent the plasmolysis of a cell, but cells do have ways to prevent cytolysis from happening. A plant cell actually has a cell wall, and so those cells can only swell up so big. Plant cells have a cell vacuole, which fills up in the water. The central vacuole fills up with water, and then there's pressure inside there. It's sort of like blowing up a balloon. And so if you blow up a balloon, think about almost blowing up a balloon inside of a box. If you blow up a balloon inside of a box, you can only blow it up so big. But then there's pressure against the outside of the box, right? If you blow up the balloon. Well, there's water pressure inside of that central vacuole, and it actually, that pressure then prevents more water from moving in, because the contents are already pushing against the cell wall. And so plant cells are generally not going to rupture because the cell wall is going to prevent that, and that extra pressure called turgor pressure is going to prevent the rupture of that cell. Now, animal cells, not all animal cells have these, but some animal cells, and we'll study some specific creatures that have these, they have what's called a contractual vacuole, and they actually have this little organelle inside of them that if they start pumping water, or water starts moving in by osmosis, they will actually pump the water back out so that they don't end up exploding. Okay, so plant cells and animal cells both have mechanisms to counteract cytolysis. Now, I do want to give you a little memory trigger for remembering the difference between hyper and hypotonic. Now, isotonic starts with a different letter. So isotonic means that everything is already balanced. In fact, if you kind of take the eye and turn it sideways, it might look like a teeter-totter. And so everything's balanced already. But hyper and hypo, how are you going to remember the difference between them? Well, hypertonic is when things start to shrivel up. And if you kind of say the word funny, you go hypertonic, and it's shriveling up because it's all scared. Hypertonic. That tends to make me think of something that's going to contract and go inside like this. Hypotonic, this has an O right here, and that O looks like a cell that's all swollen up. So hypotonic means the water is going to rush in and your cell is going to swell. So the O should remind you that it's going to swell up big, and the hypertonic reminds you that it's going to shrivel up like this, okay? So that will finish up our discussion on osmosis. You will need to use all of these terms as you are talking about what happened in your egg experiment as you're writing that up.